Welcome everyone to the introduction to metabolism. Today really marks the transition from learning about biomolecules and enzymes kind of separately to talking about them linked together in metabolic pathways. Our learning outcomes for the day are some new vocab, catabolism, anabolism, not a huge focus. We'll get a brief introduction to oxidation and reduction. We'll talk about the biochemist's way to think about this compared to the chemist's way. We'll be able to define metabolic flux through a given pathway, and we'll see how important the rate determining step is. Finally, we will talk about controlling flux, but the great thing here is we've more or less learned everything we need to know about how to control flux. We just didn't have the words for it yet. Today's Meet a Scientist moment is Emmett Chappell. He is a Black scientist, most famous for his work on bioluminescence. And we are all familiar with bioluminescence if you've seen a firefly or lightning bug, depending on where you're from in the United States. So these bugs have a small molecule called luciferase and an enzyme called luciferase and a small molecule called luciferin. And when the enzyme performs its chemical reaction with ATP, it produces light or bioluminescence. And this technology from Emmett Chappell is used today in labs to measure levels of ATP within cells. For example, I used this technology in my PhD thesis. Let's jump into it, folks. So when we're talking about metabolism, we could either be talking about catabolism, where we're kind of breaking down large molecules into smaller molecules. We're degrading these molecules to get energy. Generally, this is an oxidative process, so you'll see how these molecules become more and more oxidized. Why? Well, we want to either release energy or create energy to store. In contrast, anabolism is the biosynthesis of larger molecules from small parts. This is more what you'll learn about in Biochem 2, so we won't talk about that much together. But if you're taking comprehensive biochemistry, we will get into it a bit more for that class. This generally requires an input of energy. In this class, to give some vocabulary, that's not important. We focus on aerobic heterotrophs. We focus on human metabolism, right? So we get free energy through the oxidation of organic compounds that we eat. Think sugar, think fat, think protein. Some of the other nutrients, vitamins and minerals that we need, you'll see kind of popping up here and there as cofactors that are required for some of the reactions that we perform. Of course, we require water and oxygen, and we will see both of these come in as well at the very end of the metabolism we discussed together. What is a pathway? We're here to learn about metabolic pathways. Any metabolic pathway is a series of connected enzymatic reactions. We talked about this a little bit when ATCase was introduced. ATCase makes a precursor of pyrimidines. Its pathway is that enzyme, ATCase, and the following enzymes that work in sequence to convert small molecules into pyrimidines. In a metabolic pathway, generally we'll call any reactant or substrate, any intermediate product or products, metabolites. Metabolite is like an overarching word for any small molecule used in metabolism. There are about 4,000 known metabolic reactions in the human body, but we are only going to focus on the ones that I've highlighted with this box to the right. It might still seem like a lot. But just to break it down and to foreshadow, folks, we are going to be focusing in Biochem 1 on just working our way from glycolysis the citric acid cycle to oxidative phosphorylation. For comprehensive biochemistry, we'll go a little bit further and start to talk about the metabolism of non-sugar biomolecules. But let's start with sugar. We'll talk about glycolysis in the cytosol. We'll talk about the citric acid cycle, or the TCA, in the mitochondrial matrix, and finally oxidative phosphorylation and the electron transport chain in the inner mitochondrial membrane. We will see how this compartmentalization is important. We will see production of ATP, of molecules called NADH and FADH2. Up next, we'll talk a bit more in detail about metabolism.